Are you, are you ready to go, Dan?
I'm ready.
Okay, so um, it is my pleasure to introduce Dr. Dan LaBarbera, who's going to be giving the seminar today. Um, Dan did his bachelor's degree and PhD degrees at Arizona State University and then postdoc at the University of Utah prior to coming to University of Colorado School of Pharmacy in 2008. So Pac-12 born and bred, and all the way through. I've known Dan since he got here. He is been a great addition to the School of Pharmacy in terms of somebody that knows medicinal chemistry and some pharmacology and able to do a lot of cool stuff with novel agents. I've been working with him more or less since he got here. Um, Dan has joined what used to be the pharmacology shared resources. We've reimagined and reinvented it as the drug development and discovery shared resource. So he's a co-director of that with me now, and really going to run and coordinate all the high throughput screening and kind of drug discovery processes associated with that. So it's with pleasure I introduce him today and thank him for agreeing to do this seminar on relatively short notice. With that, Dr. LaBarbera, it's all yours.
Okay, let me share my screen. And I'll try out the laser pen function, laser pointer function. Great, and so today I'm going to talk to you about a project we've been working on for several years, and it's the development of first-in-class inhibitors of oncogenic CHD1L towards a novel treatment for colon cancer. And I'm going to reserve a little overview of the talk. I think this is a real comprehensive example of a drug discovery endeavor. And so you'll see a lot of important techniques and different models and methods that we utilize from high throughput screening drug discovery all the way through pharmacology and in vivo efficacy and pharmacokinetics. And so we'll start off just giving a little background on the pathology that we're trying to target with the Wnt TCF transcriptional pathway, the different therapeutic strategies that have been developed for that pathway, and including our own strategy. Also, the validating CHD1L as an oncogene involved in TCF transcription, driving EMT and other malignant properties. And then the high throughput screening aspect of it, leading to the first-in-class inhibitors of CHD1L, the hit to lead validation, and then in vivo studies. I'm also going to reserve some time at the end to introduce, as Dr. Gustafson mentioned, the new drug discovery and development shared resource. And this is affiliated with the Cancer Center, but also we have a new endeavor that we're launching here this spring, which is a new center for drug discovery. And I'll talk about our vision for that center and brand new infrastructure. And the infrastructure for both the center and the Cancer Center are going to be part of this new D3 or D cubed shared resource. So just to give a little background on colon cancer, many of us are aware of this, but the colon is actually a pretty interesting organ. And if you stretch it out in length, it's about 5 feet in length. And it's the major organ for removing water from digested food in the body. And as such, the colon is lined with a epithelium that secretes a mucosa. And that epithelium is comprised of here these colonic epithelium crypts, known as the crypts of Lieber-Kuhn. And these crypts are highly ordered with stem cells at the base of the crypt. And then they have proliferating progenitor cells that feed up to the top part of the crypt. And this process of regenerating cells and shedding old cells takes about 7 days on average. So if we look at a cartoon version of the crypt plays an important part in terms of colon cancer, because one of the major drivers of colon cancer is through the Wnt beta-catenin TCF transcriptional pathway. And you can see that if we follow the left side, under normal anatomy and conditions, these stem cells feed these progenitor cells, which can migrate up the crypt. Once they reach a certain level at the crypt top, they'll differentiate into goblet cells, which are able to secrete the mucosa. And this whole process is tightly regulated by the Wnt signaling pathway. And APC mutations and other mutations in this pathway, such as with beta-catenin, are initiating steps in the beginning of colon cancer and lead to colon cancer progression. And so on the right, well, actually on the left, as these cells, these progenitor cells, have aberrant TCF transcription as they move up the crypt, they do not differentiate, and they maintain that proliferating phenotype, which leads to colon polyps. And all of this is driven by as I mentioned, by the Wnt signaling pathway, and basically regulation of beta-catenin and activation of TCF transcription. So we and others have shown that TCF transcription also functions as a master regulator of EMT in colorectal cancer, and so EMT has played a major role in colon cancer in terms of tumor progression and tumor heterogeneity, and leading to metastatic dissemination. So this pathway has been a sought-after pathway for drug development for quite some time, all the way dating back into the 90s. And there's been a lot of therapeutic strategies applied to try to target the Wnt pathway, but to date, nothing has made it through the FDA requirements. So nothing really targeting this pathway directly is FDA approved yet for the treatment of colon cancer. But there are plenty of drugs and other therapies in clinical trials. And so some of the strategies are to target tankraces, which activate the destruction complex and affect beta-catenin accumulation. Others have been to target Wnt ligands, because those involve activation of the Wnt signaling pathway. And yet others are targeting beta-catenin or protein interactions with beta-catenin. Probably one of the more successful ones recently is this PRI-724, which is a compound that blocks CBP, which is also a co-activator of TCF transcription. But it blocks the protein-protein interactions between CBP and beta-catenin, leading to loss of transcriptional activity. Our approach is quite different in that we've taken an approach to target DNA binding factors. So it turns out the TCF transcriptional complex actually doesn't bind DNA with such great affinity. So the way it functions is it recruits other DNA binding proteins, such as topoisomerase 2 alpha and CHD1L, which is the topic of today's talk, to help dynamically regulate gene expression, but also activate or expose promoter sites of TCF target genes for transcription. And we've published 3 papers over the last several years in that area of therapeutic development. A more interesting discovery was published fairly recently in 2019, which showed that polyps actually shed metastatic cells, and that they were typically or thought of as being benign, but they can shed metastatic cells early on, before even entering stage 1 of colon cancer. And that TCF4 and EMT were major drivers of that early metastasis. And so therefore, therapies that may be effective and safe for both early and late stage metastasis by targeting TCF transcription may prove to be an effective therapeutic strategy. So this presentation is really going to focus on a target. And we actually chose this target through literature searching, not necessarily from screening, although my lab does quite a bit of high content screening. And when we utilize a high content screen, that is typically a phenotypic based screen, and that requires a cell model where we conduct assay development screening. We identify hits and leads from high throughput or high content screening, and then we do target deconvolution to get to our target. If we start with a target, then our strategy is target validation, assay development, screening, and then getting to our leads and hits. And a lot of us may know this, but the term content just means a lot of material, a lot of information. So high content is typically, screening is typically reserved for cell-based phenotypic screens, and it gives you a lot more information, whereas high throughput screening is typically reserved for targeted screens against a particular molecular target or process in the cell. So our first step in going after CHD1L was validating it as a therapeutic target. And there was a lot of literature reports that suggested it could be a good druggable target, but we had to do the work to prove that. And most of that work, and almost all the work that I'm going to talk about today, was recently published in Molecular Cancer Therapeutics this last summer. And so I'm not going to reference every slide, but it came from, a lot of the work comes from this work that we just published. So the first thing we did was, when we got interested in CHD1L, was we looked at the public databases, and there's this CIT database from France, and they had been collecting patient samples since the 1990s, and so they had over 20 years of patient samples. And so we started to do analysis of those patient samples in the context of CHD1L expression. And there was CHD1L expression is positively and significantly associated with a lower overall survival when we looked at all the patients over a 15-year time period. In addition, because stage 1 patients are likely to survive and stage 4 patients are likely to be deceased, we looked at stage 2 and 3, and again, CHD1L expression was significantly correlated with lower overall survival. In addition, we looked at CHD1L expression in the context of cancer stage, and we found that CHD1L expression did increase as the stages of cancer increased. Also, CHD1L expression was positively correlated with lymph node metastasis. And I'll point out too that even though the N3, which is the farthest of the invaded lymph nodes, the trend is the same, we weren't able to get significance here, and that's because the number of patients that had died in that cohort, so there was too few patients to get significance, but the trend was the same. So this indicated to us that CHD1L was driving, is an oncogene driving colon cancer tumorigenesis and progression, and is associated with invasion and potentially metastasis. In addition, more recently, such as with breast cancer, colon cancer has started to distinguish different molecular subtypes. And so we have subtypes C1 through 6 here, and their definitions are down below here, ranging from immune system down, deficient mismatch repair, KRAS mutation, increased cancer stem cell stemness, activated Wnt pathway and deregulated EMT, and then chromosomal instability. And CHD1L expression is highest in the C5, 4, and 3 subtypes, including the C5. And we were, like I said, we were already interested in the Wnt signaling pathway and EMT. And so that was very exciting to see that correlation there. In addition, it's also associated with normal mismatch repair, and CHD1L is already reported to be an important component in DNA repair. And so that data was also nice to see as well, compared to patients that had deficient mismatch repair. We also looked at patient samples from our GI tumor tissue bank, where we got samples from Dr. Messersmith and Pitts, and we looked at the various stages, 2 and 3, stage 4, and then primary tumors versus metastatic tumors. And we saw a significant correlation with stage 4 and metastatic tumors from patient samples from our clinic with CHD1L mRNA expression. So again, in multiple different patient databases, we see a significant correlation with CHD1L and malignant colorectal cancer. So through other bioinformatic analysis that we had did previously with AC-TAN and then looking at the RNA-seq data, we knew that CHD1L was somehow associated with the Wnt signaling pathway. And so we started to mechanistically investigate that. And so in panel A, what we're showing here is that we utilized the DLD1 cell line. And this is a nice cell line because it has very low CHD1L expression. And so here we have empty vector, and then we have CHD1L, DLD1 cells where we overexpress CHD1L. And then we measured TCF transcriptional activity using the well-established top flash reporter system. And you could see that we got a significant induction of TCF transcription when we overexpress CHD1L. Likewise, we used the SW620 cell line because that has a relatively high expression of CHD1L. So if we knock down CHD1L, we saw a significant decrease in TCF transcription. That was a good indicator that CHD1L was involved in regulating TCF transcription. So then we looked at first immunoprecipitation. It was already shown that CHD1L is a strong, interacts strongly with PARP1, and PARP1 was shown to be an important component for the TCF complex. So we knew there was a connection potentially through PARP1, and that data is not shown here, but it was published by other groups. But we looked at a potential interaction with TCF4, and we saw that CHD1L did immunoprecipitate with TCF4, indicating that it localizes with the TCF complex. We also confirmed that using chromatin immunoprecipitation, and we saw that CHD1L localized at important promoter sites where TCF is a known TCF target genes such as axin, vimentin, slug, LEF. We also identified that TCF was a major activator of N-cadherin, which wasn't known before that, and that was we showed that it also localized at N-cadherin as well. So then we wanted to analyze, using those two cell lines, analyze effects on EMT, because we had previously reported that the TCF complex was a master regulator of EMT in colon cancer. And indeed, when we knocked down CHD1L and SW620 cells, we saw induction of MET or reversion of EMT based on downregulation of vimentin and slug, which are mesenchymal markers, and upregulation of E-cadherin, which is an epithelial marker. Likewise, when we overexpressed it in DLD1 cells, we saw that CHD1L We saw an increase in vimentin expression, an increase in slug, decrease in E-cadherin. So these are indicators that CHD1L indeed is involved in regulating EMT. And based on our data with the TCF complex, we believe that is through CHD1L mediated TCF transcription. Likewise, effects on TCF transcription, inhibition of TCF transcription, and reversion of EMT are associated with stemness or cancer stem cell stemness. And we looked at that using the well-established clonogenic assay. And so when we overexpress CHD1L and DLD1 cells, we saw a significant upregulation or increase in colony formation. Whereas when we knocked it down in SW620 cells, we saw a significant decrease in colony formation. So taken together, we felt like we had validated our target, and we began to pursue our drug discovery efforts. And the nice thing, one of the reasons why we chose this target was because there were no known inhibitors, and that was attractive to us. But the race was on, and there was a lot of competitors out there, but it turned out we kind of beat them to the punch, and that was pretty satisfying to accomplish that. But to do that, we spent a lot of time developing this assay, and a lot of that was as a result of developing a protein expression and purification. So this is sort of just our flow chart for how we developed our assay. So a lot of effort, more than, I'd say approximately 3 years, went into really optimizing a protein expression and purification system to get to a CHD1L protein, recombinant protein that was useful for high throughput screening. Then we developed the assay, and I'll go into details in that, but it's an assay looking at the CHD1L's ATPase function. We screened 20,000 compounds. We expected to screen more, but we found a nice cohort of hits that we are pursuing, and including the ones I'll talk about today. And then we did hit to lead validation. So that's kind of our workflow. So the first step was protein purification. And you can see here on the right, we have a pure fraction, and this is the CHD1L, the what we call the catalytic domain. So the full length is comprised of 2 ATPase lobes, which are part of the N-terminus. They also extend into the C-terminus through this linker group. And the macro domain is located entirely in the C-terminus. And the macro domain is actually an interesting part of this enzyme, in that it's been shown to be an autoinhibitor of the ATPase function of CHD1L. Therefore, when the macro domain is not interacting with proteins such as PARP1, CHD1L is autoinhibited and shut off. So in order to get a more robust assay, we utilized just the what we call the catalytic domain, and left out the macro domain just to eliminate the issues around autoinhibition. But in addition, all of the CHD1L enzymatic activity requires the ATPase function. So we figured that was it's a simple, excellent readout and way to identify inhibitors. So here's our assay validation. And the way the assay works is we have CHD1L in there, but we also have a phosphate sensor. So this is a protein that's engineered with a coumarin fluorescent tag on it. So when it binds inorganic phosphate, it fluoresces. So based on that fluorescence, we're able to measure the rate or saturation of ATP hydrolysis by catalytic CHD1L, and obtain through Michaelis-Menten type kinetics, Vmax and Km. We also did some important experiments to validate the assay, and since drugs are administered via DMSO as our vehicle, we did a DMSO sensitivity study, and we found that even up to 2%, DMSO, we saw no significant effects on our fluorescence readout. And another important area of high throughput drug discovery is having good positive and negative controls. A negative control is in almost all cases is going to be your vehicle. And that is a percentage of DMSO that has no effect on your assay readout. In this case, we chose 1% DMSO. In addition, you want to have a positive control that gives you the result that you're expecting to see with a drug hit. And in this case, we used 10 millimolar EDTA, which chelates magnesium and renders the ATPase inactive. So these are a nice set of good positive and negative controls that we can use. And these controls are built into every plate that we screen. And they allow us to get a industry standard statistical analysis called Z prime factor, which is a measure of the positive and negative controls on each plate. This is a really brilliant statistical analysis that allows researchers to screen each plate. As long as there's a Z prime factor of 0.5 or higher, we can screen each plate in singlet, and that translates to a cheaper drug discovery effort. And at the bottom, we're showing here, we did a pilot screen against a pretty large 320 different kinases. And because we wanted to rule out potential off-target effects from the beginning, and this shows that we actually got no hits. So none of the known kinase inhibitors that we screened in this pilot screen had any inhibition effect, significant effect on CHD1L activity, which was good to see. So with that assay, we then screened 20,000 compounds. The top panel shows all the data and our hit limit. The hit limit is empirical. We could set it wherever we wanted to, but we decided to set it at 3 standard deviations from the mean, which gives us a 99.7 hit confidence rate. And so we're showing you all of the different hits here in this scatter plot, which we pursued and are pursuing. And then this is just the Z prime that we got per plate. And so all of our plates had a Z prime of 0.5 or higher across the board of 64 plates. And this was in a 384 well format. With those hits, we then turned to a hit to lead validation program. And this can be quite intensive and require a lot of time, different models. And so you can see here we had 6 different methods to do our hit to lead validation. We utilized our recombinant CHD1L, where we did dose responses with hits. And so one of the criteria was we were only interested in compounds or hits that gave us a reasonable dose response. Then we looked at using a 2D cell model measuring TCF activity, 3D tumor organoids, looking at the reversion of EMT, inhibition of stemness measured by the clonogenic assay, and then inhibition of cell invasiveness. Then, of course, we looked at cytotoxicity, and in this case, we used patient-derived tumor organoids. So we had over 50 hits, and that actually repeated in the primary assay. But then we looked at several other hits by dose responses, and these were some of our top leads that we were interested in. And interestingly, they have some structural similarity. Like if you look at the top 3, these are all analogs of each other. And that's always nice to see when you're doing high throughput screening, because that shows you that similar structures are working similarly. So in some ways it's a hit validation in itself, or validates the assay. In this case, these top compounds were pretty potent inhibitors of CHD1L, but they were deprioritized later on because they didn't show activity in cell-based assays. On the other hand, compounds 4 through 7 were still potent inhibitors of CHD1L, but they also showed potent activity in cell-based assays. So we then looked at the next step was TCF transcription. And from these experiments, compounds 5 through 7 emerged as our top leads that we wanted to pursue. There's nothing wrong with compound 4. It just showed less of a dose-dependent effect in this cell-based assay. But that led us to prioritize compounds 5, 6, and 7. Now, we turn to our EMT assay, and let me explain this a little bit. So one of the things we did was, this is using a tumor organoid model. So we culture these as single tumor organoids. They can be roughly about 750 micrometers in diameter. And these tumor organoids are so we developed a dual reporter system for EMT, where we were measuring with one plasmid, we transduce cells that measure vimentin promoter activity, as well as E-cadherin promoter activity. That way we can measure in real time the plasticity of EMT on these tumor organoids and other cell models. And we can actually isolate these and plate these isolated phenotypes. And I'm not going to go into detail about it, but it hopefully will be coming out in the new Oncogene paper in 2021. But this is how we culture the organoids, and we use a 3D high content screening analysis method to measure the changes in EMT using our dual reporter. And the way we do that is we'll take optical slices through the tumor organoid, such as this. And the beauty of this method is it's actually fairly fast compared to doing volumetric analysis, which can be done, but it's not high content. You can analyze effects in a single slice, or you can summarize these in what we call a maximum intensity projection, and then analyze the data in that maximum projection. And so that's what we did. And I'll show you what the assay looks like in real time. This is a time-lapse video. And compound 7 on the right is not the compound 7 that I'm talking about. It's one of our topo 2 inhibitors. But you can see on the left, we used isolated GFP positive. So these are a quasi-mesenchymal cell phenotype that is, interestingly, is significantly higher malignant properties than the E-cadherin positive or the red phenotype when we isolate them and characterize them separately. But this just shows you the real-time effects of our compounds in reversing EMT or inducing MET. So to show you that data with the compound, the CHD1L inhibitors, so showing in panel A, that is, hit 5, 6, and 7. And you can see we get a dose-dependent downregulation of vimentin with a dose-dependent upregulation of E-cadherin with all 3 compounds. And just showing that with a representative images of our EMT assay, you can see as we increase concentration, we get downregulation of vimentin on the bottom with upregulation of E-cadherin activity. So this is further evidence that these compounds are acting via the mechanism that we hypothesized. We then looked at CHD1L inhibitors in terms of their ability to inhibit clonogenic colony formation and invasion. And we also looked at their ability to, using different markers other than vimentin and E-cadherin. So we looked at ZO1, which is an epithelial marker. And in this case, we treated with 5 and 6. You can see we had an upregulation of ZO1 and a downregulation of SLUG, which is the mesenchymal marker. So indicating a reversion of EMT. And then here we, 5, 6, and 7 showed dose-dependent effects on inhibiting colony formation in not only CHD1L overexpressing DLD1 cells, but HCT116 cells. And likewise, we saw a significant inhibition of invasive potential. And in this case, we used a wound assay, but rather than a migration assay, we coated the wound and all the cells with 50% matrigel. So the cells have to not only fill the wound, but they have to migrate through the matrigel to fill the wound. Then we looked at, so CHD1L and EMT are associated with programmed cell death. So the mesenchymal cells are inherently resistant to programmed cell death, which allows them to detach from epithelial sheets and invade and migrate to distant sites. And so we looked at, here's a time course where we looked at E-cadherin promoter activity, so RFP activity, in relation to DMSO. So within early hours, we see upregulation of E-cadherin or induction of MET. And that induction of MET was prior to the induction of apoptosis measured by using a reagent called cell-tox green. And then we looked at, so cleaved E-cadherin is known to activate apoptosis via the death receptors. And we saw with compounds 5 and 6 an upregulation of cleaved E-cadherin based on Western blot activity. And that also correlated with increased cleaved PARP and cleaved caspase 3, which are markers of apoptosis, as well as cleaved caspase 8. And then, of course, we confirmed the apoptotic activity with an exon 5, which is a pretty well-established marker for apoptosis. And then finally, we showed that compound 6 had a pretty good cytotoxicity against a patient-derived tumor organoid of colorectal cancer, shown here in panel E. Right. So CHD1L, as I mentioned earlier, has also been reported to be an important component of DNA repair, in particular PARP-mediated DNA repair. And so we looked at CHD1L inhibitors and their ability to target, enhance DNA damaging chemotherapy activity. So we chose SN38, which is the active pharmacophore of irinotecan, which is one of the standard of cares for colorectal cancer. It's a topo-1 inhibitor that causes DNA damage. And interestingly, if we use parental DLD1 cells, which remember have low expression of CHD1L, we didn't see any synergistic effect with our compound. But when we took those cells and we overexpressed CHD1L, we did see a measured significant effect with compound 6 and SN38 synergizing to increase the amount of DNA damage measured by gamma H2AX. And so this assay is really nice because it shows, one, on-target effects of our compound, our lead compound, and the data demonstrates that potentially CHD1L, in addition to affecting EMT and TCF transcription, may play an important role in the DNA repair mechanisms, which it does. It's been reported in other cancers, but not necessarily, it hasn't been reported in colon cancer yet. So then we started looking at synergy with respect to cytotoxicity. And in this data, we're showing SN38 alone, which are the orange data points. And in that case, we had a 300 nanomolar EC50 for cytotoxicity. And then SN38, or I'm sorry, 6 alone was 5,000 nanomolar or 5 micromolar. 6.3 is an analog of 6, and I'm not showing the structure here, and it wasn't shown earlier, but it is an analog of 6, and it's more potent, 3 micromolar. And so with 6 and 6.3, which are, again, derivatives of each other, we saw a significant synergistic effect with SN38. And this translated to enhanced potency of 50 to 150 fold compared to SN38 alone, which is, as far as I can tell from the literature, is the most potent synergy with SN38 that I could find. So of course, the next step was to look at our one of our lead compounds. In this case, it's compound 6 in vivo. And we did this in collaboration with Dan Gustafson. And so we sent him our compounds and the samples, and he measured the initial PK activity. And here are all the important PK parameters. And notice the half-life is 3 hours. And what I want to stress to people doing drug discovery is I recommend you get as soon as possible into PK studies, because it really determines how the efficacy is going to turn out, because you can really design your dosing studies accordingly. And this really helped us. So because the half-life is 3 hours, we decided then when we did an animal study, we decided to utilize a 2-dose-a-day schedule 7 days a week. So in this case, we did an in vivo efficacy study looking at tumor volume. So black data points are vehicle, red are compound 6, the CHD1L inhibitor, orange is irinotecan alone, and then the green is the combination of 6 plus irinotecan at 5 mg per kg for 6 and 60 mg per kg for irinotecan. And as you can see, we saw a significant positive inhibition of tumor volume, almost down to 0 with the combination compared to irinotecan alone. And the statistics are shown here in this table. So the combination of irinotecan and compound 6 significantly inhibited colon SW620 tumor xenografts. And this was in a 28-day time course. So with that, our future development. So this cartoon on the left basically shows that if we can inhibit CHD1L mediated malignant gene expression, but also its involvement in DNA repair, that we have an effective therapy against drug-resistant malignant cancer. So our plan is to expand these studies, and we've been working diligently on this, and I can tell you we've made huge progress in the medchem area, and that'll be something we'll report later on. And so our goal is to move to studies that will allow FDA enabling studies to get to an investigational new drug, with a goal of trying to get this moved to the clinic. So this is the most important slide, because this is all the people that did all the work, and everybody here has touched this project, but I want to give Josh a special acknowledgement. This was his actual PhD dissertation, and so a lot of the work was done with Josh leading a collaboration with many of us, and then Chung worked really closely with Josh on that, and Hector as well, and Laura led all the animal studies with support from Tiger. He goes by Tiger, but his name is Shin He Lai, and also Travis Berneski. And then also MedChem in the area of Brett Pregaro. He's a PhD chemist in my lab, and also Abraham, who received his PhD with me, stayed on as a postdoc for a year or so, and now he moved on to Eli Lilly and Company. And then all the others here who helped us out. And Brett and Jason are now, Jason Gregler are working on the MedChem aspects. And also collaborators. We had some really great collaborations in this work. The Messersmith lab, the Todd Pitts lab, Dan Gustafson, Dan Reagan, who works with Dan Gustafson. He did our pathology. He's a DVM over at CSU. And then Uday Campello recently has been helping us with some microsomal work on some of our analogs. And then funding for this work. So what I'd like to do next is introduce this new Center for Drug Discovery, and then if we can reserve time at the end for overall questions on the seminar, but also any that you might have on the Center for Drug Discovery, and the new shared resource. So we've been working on this endeavor for a couple of years at the School of Pharmacy, and I want to thank, before I get going on this, thank Dr. Ross, who's the chair of the Department of Pharmaceutical Sciences and Associate Dean of Research and Graduate Affairs, and then also the Dean Altier, because both of them were very supportive of this endeavor and helped raise funds to start establishing this. And the vision of this new Center for Drug Discovery is to really lead a grassroots initiative beginning with CUAMC and expanding outwards in Colorado to other institutions, academic institutions, and also local industry. And we want to innovate drug discovery through collaboration. So we want to innovate through collaboration. And this is our what we call our drug discovery collaborative hub diagram. We intend to collaborate heavily with all the different established centers and institutes, schools and departments on campus and throughout Colorado to make this a huge success. So our goal is to establish a center. We're already working diligently on bringing it formally together with the paperwork. The goal of the center is to bring researchers together to enhance drug discovery on campus. We want one of the biggest assets on our campus is all of us, all of the labs on our campus and through the different campuses and throughout Colorado have unique disease models or unique patient samples that can be harnessed for drug discovery. And we want to help investigators harness those resources in their labs and to help them utilize that as a drug discovery tool. And then we want to collaborate with existing centers. One of the key partners we want to work with is the Cancer Center, and we envision that we'll be able to help bolster the developmental therapeutics program through this initiative to develop more in-house cancer drug discovery here. We're going to work with the CCTSI and the CCPN to utilize translational and precision approaches to drug discovery. We want to try to help provide some education and training in drug discovery as well. And a really important component will be working with CU Innovations to just build intellectual property here on our campus that can turn into products that can be translated to the clinic or licensed to companies for further development. With that said, we have to have the right infrastructure to accomplish those goals. And that is where this initial investment came from, primarily from the ALSAM Foundation, but from other funds from the university. And that's to obtain this new equipment and infrastructure. And this is the G3 Explorer workstation from Perkin-Elmer. And so the picture's small, but it's actually a fairly large apparatus, 15 feet wide by 8.5 feet high by 8 feet deep. And this is going to house, it's a HEPA filtered enclosed system that's going to house all of the automation and all of the instrumentation for high throughput screening, including the compound library storage is right here, a new Phoenix Plus high content imaging system, and many other equipment that are shown in this blueprint schematic. And what's really cool about this now is we've had some of this equipment. We have a Phoenix now, we have a Janus, but it's not integrated with a robot. And so there's still a lack of automation here. In this case, our system is going to have this robot, which can access both the top deck and the middle deck. And I should point out there's 3 decks here. There's a top deck with the main equipment, and then a middle deck that has other equipment, and the bottom deck is mostly storage. But the robot is on this track system, so it can access and integrate with all of this equipment on the top and middle deck, essentially increasing the rate of high throughput screening. And in general, there's no reason why it can't screen 24 hours a day, 7 days a week with this robotic capability. So this infrastructure is going to be maintained and utilized in and is affiliated with the Center for Drug Discovery, but also is affiliated with the Cancer Center. And the Cancer Center is supporting this. And so there's going to be a discount for this for Cancer Center members. And all of that is going to be maintained in this new drug discovery and development shared resource. And so this is, as Dr. Gustafson mentioned in the beginning, the merger of the in vivo pharmacology core that was existing and our previous high throughput screening core that was existing. And it's also bringing together other core facilities on campus that play a major role in drug discovery, such as Mike Wempe's medicinal chemistry core and Phil Regan's computer modeling core, and other cores that are doing drug discovery, such as mass spectrometry or NMR. And then Dan Regan is also involved with, he's been heavily involved with working with us in the area of pathology and toxicology. So he'll play a role in this as well. So with that, that's all I have. I'm happy to take any questions at this point. It's hard for me to tell if people are still out there.
Hey, Dan, this is James Gregory. I had a question. I put something in the chat, but you're right. I don't know if you want to stop sharing your screen. Otherwise, we're getting a giant black box. Oh, that's good. Yeah, maybe go back to one of those sites. But actually, first off, I think these new facilities are incredibly exciting. So I really look forward to seeing what you guys can do with it. But going back to your talk on CHD1 or 1L,
yeah.
Your in vivo efficacy is quite impressive, but I wonder if you've actually already anticipated the development of resistance. And, in other words, they even did this with BCR-ABL when they first came out with, imatinib and Gleevec. They, by using screening, they actually identified tons of mutations that could give resistance. And indeed, many of those ended up being found in the clinic, as the years went by. And because that would serve two purposes. One is you could actually anticipate resistance, but on top of it, it would give you a great understanding of off-target effects. Because if you get a resistance mutation and now your drug doesn't do anything to the cells, your drug is essentially all on target. So I don't know if you're doing anything in that space.
So we're just at this point characterizing the synergy with the cell models we have and in vivo models we have. But I think you raise a great point. There's a lot of unknown here that can be explored. And, certainly for us to do more mechanistic areas, we need to collaborate with, faculty like you and others who really focus on mutations and things like that. So that's something I'm interested to collaborate with people on. And that would be a great addition mechanistically to our understanding of CHD1L mediated DNA repair. A recent paper came out that showed that CHD1L is the major factor causing drug resistance to the PARP inhibitors in the clinic. And that was literally just published like a month or so ago. And they actually referenced our molecular cancer therapeutics paper saying that CHD1L inhibitors from here should synergize with the PARP inhibitors. So
right.
That's
so
something
your drug could have could have efficacy beyond colon cancer.
Oh yeah, in fact, CHD1L is implicated in lung and breast cancer, and also it was discovered from liver cancer. It's also called ALC1, amplified in liver cancer 1. So that sometimes confuses people. We kind of stuck onto the CHD1L, but it's also called amplified in liver cancer 1.
And I think Mercedes has a question too.
Yes.
Hi. Hi, this is Mercedes Rincon. I'm in the Department of Immunology and Microbiology, and I'm relatively new here, but this was really informative for me to know about this core. And I have a couple of questions. One is, in your design, do you include also the effect, testing the effect in normal cells? Because one of the major problems of all the drugs eventually is that they're toxic. They kill the cancer cells, but they kill normal cells. So,
absolutely.
And I wonder if you have like lymphocytes, human lymphocytes, something that are not cell lines that you can test the effect of your compounds?
So we don't have human lymphocytes, but if you have them, we'd be happy to test them. But what we did get was from ATCC was normal colon,
Okay.
and we got some other normal cell tissues, like primary tissues. And I can't say for sure with the CHD1L inhibitors, but I can tell you we've tested other compounds that have a similar mechanism of action with our topo2 inhibitors, and we don't see the effects that we see in the tumor cells that we see in the normal cells. But we're currently investigating that with CHD1L inhibitors as well.
Okay.
Dan, Raj here. So I'm following on Jim's question. So it's not only the resistance, how you answer the toxicity once you start combining these with the real drugs. I mean, the data you are showing in this curve with a combination almost complete inhibition, which is great. But
Yes.
if you follow it up, I mean, how toxic will be that combination?
Exactly. Well, the point of the combination is we're hoping is that we can significantly lower the dose of the cytotoxic compound, such as irinotecan. So it turns out people, I'm not a clinician, but I've read some things that people can actually die from this type of chemotherapy. It's pretty toxic. And what you'll notice here is actually at 60 mg per kg, it's still, we feel like this is actually still too high. And so in our next animal study, we're going to try to drop that dose. So our goal is to show, and as we get more and more potent inhibitors, which I can tell you we do have more potent inhibitors, I just can't talk about them yet, that we feel like we'll be able to drop both CHD1L dose concentration, but also the chemotherapy. And I have other data that our lab has generated that show it synergizes with other DNA damaging drugs like etoposide and oxaliplatin. So
how was the, excuse me, how was the toxicity in this experiment?
So the toxicity, the animals were fine grossly, and we just finished harvesting all of the animals, and so we intend to look at liver toxicity.
Right.
What I didn't talk about, we already looked at acute liver toxicity with the CHD1L inhibitors, and there was absolutely no toxicity. And Dan Gustafson did that work in collaboration with Dan Reagan.
Okay, thanks, Dan. Solid stuff, obviously. Yeah.
Thanks, Raj. Great questions.
I mean, I think that the key is in these combinations is going to be looking at, you know, the gastrointestinal epithelium, certainly with irinotecan, and making sure you don't, you know, sensitize that, because that's the major toxicity associated with the camptothecins.
Right. Which led to the prodrug, right? So.
Yeah, absolutely. So I think that, you know, those studies are certainly in the books. But
well, we didn't observe any toxicity at these doses in the animals grossly, but we were just talking about that lab meeting this morning. Should we harvest those livers or organs to send to you?
I think you should harvest them. That's a hallmark of.
I don't know if Laura's on listening, but Laura, we need to harvest those.
Yeah, have those tissue harvested, and we can talk offline if you need any help. I can help you because we do all the time these studies.
well, I wouldn't look at liver. I'd look at gut.
Okay.
I'd look at,
So just
I think you can
have those tissue harvested, because you know once you harvest and store them, you lose nothing. But if you need them and you don't have them, then you lost your whole experiment to do again. And then you do at least H&E staining and compare the normal pathology and things like that. Those are important things to make your point, and that's why I asked that question.
I think it's
I
another.
have to go for another meeting. Sorry. Great job, Dan. Thank you.
Thanks, Raj.
So instead of the liver, you can also collect the serum and looking for like ALT, AT, AST, which are liver enzymes.
yeah, but there's no reason with this drug, because I don't think you're going to see liver toxicity.
Oh.
I mean, it's much more likely.
Yeah,
GI.
we already looked at the acute liver toxicity over 5 days at the maximum tolerated dose. We didn't see any.
You didn't see anything, yeah.
Yeah. I guess we need to do chronic dosing, so we didn't look at that yet.
Yeah, I mean, some of that comes out in the wash when you do the therapeutic studies, and you just have to be, you know, smart enough to collect the right tissues. And, you know, I get that you know, what you see in a mouse is a lot more tolerated than in people, but if you do it right and you look at crypt depth and proliferation of the you know, the GI epithelium, you can actually get a pretty good clue in terms of what's going to happen with this stuff.
By the way, there's a couple questions in the chat.
Okay, I'm not good at monitoring chat rooms.
I could look at it right now. One is from Zero Wang. It's how did you express your CHD1L in E. coli? All right, did you express it in E. coli? Was it in E. coli?
Yeah, we expressed it in E. coli.
Okay.
So we bought the expression vector and expressed it, and maybe she has more specific question about it.
Yeah, I don't know. If the person wants to ask more, I guess they can chime in. But the other questions from Paula.
Also, if they want to go look at, I cited the publication.
Ah, right. Yeah.